提供比较。
And some you do not have this label, so I'm confused with which one. Okay, this is the figure arrangement. So, and I have another question on your data, table 4. Can you find the data, table 4? And for your MBC, you have around 100, and MBN, you have like 50 or 60. And if you calculate the ratio of CM ratio, you have like less than 3 or 4. I do not think it's right. Yeah, you may need to check the data. Like for MBC and MBN ratio, we only have at least 4 maybe. Or the soil was sterilized. You sterilize the soil? I'm not sure. And you also have the DOC was quite low. It's only 2 less than 3 ppm. It's so low. And you already I think you pre-clipping, but you already cultivated the plants. So why the DOC was so low? Just to check the data. Okay, these are data. And another one more concern was on your, you say when important find your structural equation modeling, right, for figure 5.3. And in my opinion, I cannot understand. In my opinion, you have clipping treatment, and then clipping will affect plant growth or plant community composition and their competition, and then will affect carbon allocation to the ground, and then will affect microbial growth, and then like for example, some species increase, some species decrease, and they will affect enzyme reduction, and then this enzyme will affect carbon sequestration or nutrient cycling, right? So yeah, I do not agree with this one. So yeah, these are all my comments. So thank you. Yeah, I learned a lot. Congratulations, you did a very good job.
Thank you so much, Professor. Thanks for your genuine question that you asked. Thank you for your precious suggestions. So you talk about the title, so we will revise it, and I will also discuss with my professor about the title in further detail, and I will change flowchart as you suggested me. As you told me that you go for data quality and control, so I will see also this, and also I will include the seeds ratio more detail that we measure in the lab. That we actually use the 30 gram for dactylus and 30 gram for lullium and 40 gram for a legume. So, but we measure it and then we deliver it to the field and then we sold it. And for plant sampling, I will also include some more detail to make it simple and easy. And figure 3.5, I also separate figures to deliver it easily to the readers, and also I will see the MBC and MBN values and ratio, and also I will review your suggestion about figure 5.3 SEM. Thank you for the suggestion.
Thank you.
Thank you.
好，请张老师看看有啥意见。
Hi, Safar.
Thank you.
Congratulations. I've reviewed the professor show. There I have some suggestions. First, about the title. I think you should add the 3 species. All at the areas. Yunnan province or Yunnan Guizhou planter. Second, about the abstract, there's some details about the Chinese abstract. I think in the second paragraph, I think the area is the subtropical area, not the temperate areas. Third, about the third point in the chapter 2 about methodology, I suggest you complete the some study set, such as the annual average temperature, annual precipitation, such as these details. The third about the conclusions, I suggest you should shorten the conclusions just like your report. This is my suggestions. Thank you.
Thank you for question. So as you suggested that we should need to include 3 species and also include the Yunnan Yunnan as a place. So we will include it, and also that we will include the annual precipitation and annual temperature in chapter 2, and also we will include your other suggestion. Also, I will short the conclusion.
Okay, thank you.
Thank you.
Congratulations.
Thank you.
好，请孙老师。
Conclusion said that and I agreed with the product chair and I think in the field you should deduct the brand computation. I suggest I understand your
孙老师，
is
孙
right.
老师，你的声音这个忽高忽低啊！你是在办公室还是在里面啊？
对，我是在办公室，我没有玩吧。
啊，我这听你声音一下高一下低，可能没怎么听清楚了。
那我再重新。
好。
这个I suggest you select the brand computation. Can you listen clear?
更不清楚了。
I understand. 听说。
Can you listen? Can you hear me?
Is that right?
Repetition. Sorry, teacher, I did not hear you. I'm not listening.
I agree with the professor and still you suggest you select the as competition from your tail. So the second in your abstract, I suggest you conclusion the summary of results from the community, so you may look at the act of recording. In the introduction, I think you should understand the clamping is different from the glacier. The glacier will choose of the food what they like to eat. So but for the clamping, there was no choosing. Do you understand this? They are very different. So in the introduction, I think you should, I suggest you understand the difference. I have after the suggestion, I will send you later. Okay?
Okay.
That's all. Conclusion table.
好，谢谢孙老师。请看看刘老师还有什么意见。
Hello, Steve.
Hello, teacher.
I have 3 main concerns and a few suggestions.
Okay.
So first of all, let's look at your title. You said you mainly focus on 2 factors, sleeping intensity and plant composition, right? For the first one, how do you set the clipping density? What I mean is, do you try to simulate the people who are doing it in the nature, or you just set this density by yourself or with your supervisor? Can you hear me?
Yeah, okay, boss.
Mm-hmm.
So,
The secondary, yeah, we want to reply first.
and density. We have
Right.
some density and density.
Why did you set this for clipping treatment?
Clipping?
Why set you such for treatment?
Okay.
Yes.
Okay, Professor. Actually, many research that I study, they used 10 to 7, 8 clipping intensity. So in high clipping intensity, I cut it 8 times because to keep it high disturb level. And medium, because in medium after 3 months, so it's like look like normal that after 3 months you need to give some further stock to your livestock. So for 3 months you also need to store some fodder for the livestock. So I keep that 3 months. But the light one was just like we keep it just like very light, just 2 times. And control was so that's it's like we use like consider it like annually and according of the livestock production. In medium we use it because that is like look like suitable to give fodder to the livestock. But the other one is like very high one, but we use like for just to get very high disturb level to disturb the system, that how they respond and how the plant adopt the strategy to this stress resilience and how they adopt the ecosystem function for survival and competition. So that's why we in high clipping we use like a much more stress to the ecosystem.
Mm-hmm.
And about the density of plants, as you know, Professor, like more than 50% of grassland they represent graminide. More than 50% grassland they have graminides, and other like 20 or 30% they have legume. So actually our ratio represent the grassland ecosystem all over the world. The maximum grassland they represent graminides, and they have 20%, 30% legume. But in our we have cultivated model, so just we consider one species. But in future, if anyone doing, so he can consider any other species together with this species, because every species has own strategy. Like my tripolium legume species has creeping strategy. They're going to creep on the land, on the surface of the soil. But maybe other species, other legume has different growth strategy. And similarly, like lolium and dactylus, they have they increasing tailor numbers and producing bud production after clipping. So you know it also depends on the plant growth strategy. So maybe if anyone doing in future, so I recommend that they consider different species composition. They have different growth strategy, and they should consider its effect on the ecosystem. Maybe they find much better results.
Mm-hmm.
Yeah.
Actually, what I want to say is, you should find some support for this design. You know, in nature condition, in rainy season, plants grows so fast, right?
Yeah.
So no matter which clipping intensity, you should find the support or find a reason for you to do this, and this will help you.
Yeah, Professor,
to-
you are right.
Yeah.
Your question is very genuine question. As you mentioned that you need like growing season. So actually, in dry period, like we have in Yunnan, we have from November to May, but we did not do that much clipping. We mostly we did our clipping in growing season. Maybe just we did 1-2 clipping in dry period, because in dry period if we cut it many times, so maybe it will go to die, because due to cold weather, and also they are also like semi green, so maybe they will go to die. So that's why in dry period mostly we did not use that much clipping. Maybe we just used it to complete our design, just 1 or 2 clipping, but mostly like with 80-90% clipping we focus on growing season, because we have growing season from May to November, and also we have first growing season. In our study we have 2 growing season. Yeah, so
Yeah.
we mostly implement our clipping in the like 80% it's go for in the growing season.
Yes, that's good.
Thank you.
For the second thing is about plant competition. But in my opinion, it seems like you are adding clipping intensity on different species composition, right?
Yeah, actually, Professor, for competition, we measure each species like root length, for below ground competition. So then we consider each species abundance in each plot. Then after that we sum it, the 3 species. Then we have 3 species, then we divide it on 3 to get the final competition score. So because that's one of the correct way to do it. And one point is that actually why we are doing clipping. The clipping is actually, Professor, decreasing the above ground competition. It is actually because when you cut the grasses, so you actually decrease the photosynthetic efficiency, and also you give space to other slow growing species. So it's mean like you actually, the clipping is actually the indirectly is like decreasing the above ground competition. But what after we did the clipping, but after that we see that how the above ground competition that we decrease during use of clipping, so how they push the below ground competition, and how below ground competition in turn responds to above ground competition, and how they boost the community structure. So actually previous studies, they just revealed that clipping negatively affect the biomass production, but they did not tell us that when you cut the above grasses, so actually you decrease the competition above one. So what happened to your below ground competition, and how your below ground competition respond to above? Actually, these previous studies did not did it. That's why we don't know. Always we have controversy about clipping. Some said it's positive, some said it's negative. But I think the study that revealed negative, because when you consider different species and you cut the above ground grasses and decrease the photosynthetic efficiency, so it may increase the below ground competition, but I think the species combination and other studies, they did not push well the above ground competition. That's why they get the decrease in biomass.
Yeah,
I think that's why.
Yeah.
Yeah, bro.
So in general, in this thesis, it can include the 3 factors: clipping intensity, plant competition, and species composition, right?
Yeah, actually, Professor, we focus on the plant competition, above one and below one, not each species competition. Actually, we consider each plant, and then we consider together the community like competition above one, how these 3 species compete in the plot. So that's why we consider this like. So as we can, there are different methods like symmetrical competition, asymmetrical competition. So but we did not go because it was outside of our design. In our design, I think we consider the above and below one that's better represent our design.
Yeah, but I suggest you to consider about what I said, the third factor. Yeah, anyway, now I see your point. The second one is about, you mentioned the about competition and below competition, right? I know how to quantify above ground competition, but how did you do for the below ground competition?
Professor, we made below ground competition that we actually use hand shovel to excavate each species very carefully, and then we intake the root length, and also we measure the adventitious roots and lateral roots length in centimeter through ruler, and then we make it average. But we use just 20 individual of each species, and then we use that average of 20 individual of each species to divide it on the abundance of each species in each plot. So we get that below ground competition in each plot.
Can you tell the roots? Can you separate the roots by a species?
Sorry, I did not listen.
I mean, when different species going together, it's easy to tell the above ground part. But can you separate the roots by species when they growing together?
Yeah, actually, Professor, we consider like 20 individual randomly from the plant, from each plant. Each species, like 20 individual from trifolium, 20 individual from dactylus, and 20 individual from lolium. So after that, we measure each one root length as well as adventitious and lateral all. So then we have already the abundance of each species in each plant. Then we divide that each species that we got average from 20 individual of each species. Then we multiply it with abundance, and then we sum it. So we did this slide.
Okay. Yeah. The last one is about you did this work on one site within 2 years, and you want to see the effect of clipping intensity and plant competition on both soil and ecosystem functions, right? Do you think with one site only within 2 years, it is enough to support this thesis?
Yeah, Professor, you know that every research has some like shortcomings, and no research has like complete. So, but you know, like we did our hard work according of the Yunnan climate, but it is one of the first significant step towards like some future sustainable applications in the future. But maybe in the future, the other futures will do it much better. Maybe they consider 3-4 graminide species. In some they consider 7 graminide species with different levels of legumes, and also they consider maybe 2, 3, 4, 5 legume species. So every study has some shortcomings, and every study and not research in this one. Any research, you know, that is not complete. So I think I recommend if anyone is doing this further, so they can do it. Yeah.
Yeah, I have a few suggestions. You have some errors in the reference. You should pay attention to that, especially on the name of the some journals, like on page 144, 146, 147. And the second one is about there are some figures. Let's take the page 70, figure 4, for example. In these bars, they have no difference, but you labeled them as significant difference. I don't know how this happens.
Yeah, it's a very good question, Professor. But actually, my professor also asked me that. Then I rechecked it again. But actually, that you asked the question, as you know, they have p-value like 0.10, 0.16, 0.15, 0.12. So it was less than p, more than 0.05. So actually we did not consider it in our main text. But it looked like that. From labels you can see that it show like significant, but it's not like it's p-value like between 8 to 16. So it looks like yeah.
Yeah, but you should find a way to reduce this confusing feeling, right? Okay. The second one is about, in some paragraphs you write quite a lot of words, but actually they should be cut in short. For example, the abstract for each chapter, you use the 2 paragraphs to demonstrate this result. Actually, you should cut them in 1 paragraph. That's enough. Or else it looks like not that professional, I think. Yeah. And the last one is about, I saw on your PPT you said you have already published several papers, but the 2 are published in the foresty areas, not that related to what your thesis described, right?
Yeah, Louis.
I'm concerning how this publication could support your thesis.
Actually, now, Professor, we submit 1 paper in Journal of Ecology from this project. So maybe in 4 or 5 months it will be published. So from our Daslet project, we are also publishing paper. But as you know, I did also work in forest. So we have like, I'm working on 2-3 disciplines, like here in grassland also, Professor. We learn about our field work in grassland, and also I have some like knowledge about forest, so I did some publication also in forest in the guidance of our professor.
Okay, yeah, but just wondering about how this happens. But anyway, I can see the value of your thesis. There will be some good paper published in the future. Good luck.
Thank you.
That's all. Thank you.
Thank you so much. Thank you so much for joining us.
好，谢谢刘老师。I also agree with the comments from the other professors' comments, especially the title. I mean, the title should focus on the impacts of clipping intensities. I don't think plant competition is a real experiment treatment. Also, soil and ecosystem function are not clear for me. In my opinion, changes in soil properties are also related to ecosystem function. The second question is about the results in your abstract. I mean, you don't need to introduce your findings according to the contents from every chapter. I mean, you can integrate these findings. Also, I think your table 1 and figure 3 should be moved to chapter 2 because I think they are methods. Okay, I just these suggestions for your consideration. Thank you.
Thank you, Professor, for asking your suggestion. We will consider the suggestion about title, and also you are right that our main focus is clipping. So we, in our title, we mainly focus on clipping intensity and ecosystem function. Actually, we write about just like for it present over all ecosystem function. And yeah, Professor, you are right that we keep our abstract lengthy because actually our school need like more than 2,000 words, and Chinese words it's like more than 3,000 words. So that's why we keep it a little long to keep the criteria for our school. So that's why. and I will also check the figures that you recommend me. Thank you.
Okay, thank you. Thank you for your presentation. You can communicate with Professor Wu and make changes accordingly. Thanks again.
Okay, Professor. Thank you all.
好，那么下一个是我看下一个是
Clear yours.
Java 的。
那是鼠标，那是激光笔。
好的。
颜色。也是焦点吧？它是激光笔的啊。
Good afternoon, professors, and good afternoon, everyone. My name is Jawad Ali Shah, and I am from Pakistan. Today